Today we are going to talk about DNA a little bit more, and we're actually going to be talking about how it's actually useful for us. So DNA, we know it carries our genetic information. It's making us who we are, but really, how is it doing that? So today we're going to start learning about that process, which is protein synthesis. So for the function of DNA, we know it's a nucleic acid, which means its function is that it's carrying our genetic information. The DNA is always kept safe in the middle of all of our cells, which is called the nucleus. That's where it stays. On the DNA, there are certain sections that are called genes. And these are the actual important parts of the DNA that are actually used as the directions to make the proteins. So DNA can really be thought of as the recipe book to make proteins. They make all of the proteins that are in your body. So again, DNA, it's kept safe in the nucleus so that it's protected because it carries the directions to make our proteins, so we have to keep it very safe. But the proteins that our body are making using these directions, the proteins are made at ribosomes, which are way out there. You can see they're towards the edges of the cell, and the DNA is kept in the very center of the cell. So if DNA is used as the recipe book to make proteins, but it is stuck in the nucleus, how is it used at the ribosome to make the proteins? The answer is that it needs a special messenger to help it carry its message out there. And this special messenger is called messenger RNA, which we just abbreviate as mRNA. So DNA is kept in the nucleus. It gets rewritten into messenger RNA, which is called mRNA. And then the mRNA is allowed to leave the nucleus, where it goes out to find a ribosome. The ribosome reads the messenger RNA, and then it uses those directions. The ribosome uses the directions to make a protein. So DNA needs RNA. And they are both nucleic acids, so they're very, very similar. You do need to be able to compare and contrast these two nucleic acids. They have a lot of similarities. So they're made out of nucleotides. They have the phosphate, the sugar, they have the bases, but the sugars are slightly different. So in DNA, we say it's a deoxyribose sugar, and then in RNA, it's a ribose sugar. And then for the bases, we know that in DNA, there are A's, G's, C's, and T's. But in the RNA, there's A's, G's, C's, and U's. So that is a big difference between these two nucleic acids. So this little Venn diagram here summarizes the similarities and differences between these two polymers that you need to know. So DNA, we know it has two sides. It has thymine in it. It has deoxyribose sugar. It's kept safe in the nucleus. RNA, it's just single-stranded. So it's actually only one side. It's half the size. It contains uracil. That's what the U stands for, instead of thymine. The sugar is ribose sugar, and it can be found in the nucleus, which is where it's getting the message of the DNA. And then it leaves the nucleus and can go out of the nucleus into this gooey stuff that fills the cell, which is called the cytoplasm. And then that's where it goes to find the ribosome. In common, they're both made of nucleotides. They both also have the A's, G's, and C's. So those other three bases, they do have those in common. There are other types of RNA. So we're talking about messenger RNA. That is used to take the message of the DNA from the nucleus out to the ribosome, which is where it's used as directions to make a protein. The ribosomal RNA, which is rRNA, this is actually forming the structure of the ribosome, which is just like a protein factory. So ribosomes, that's the protein factory. That's where the proteins are getting made. And then the last type of RNA you need to know is tRNA, which stands for transfer RNA. We're not going to talk about this today, but what it ends up doing is it brings in the monomers of a protein, which you should know are amino acids. It brings those into the ribosome where they can be hooked together to make the proteins. So again, all three types of these RNA are used in the process of protein synthesis. They're all very important. And protein synthesis is made up of two steps. So the first step we've already really gone over. It's transcription. That is where the DNA gets rewritten into mRNA. And so the mRNA then just carries the code of the DNA from the nucleus out to a ribosome. And then translation is the second step. This is where the protein is actually getting made. Amino acids are brought in on the tRNA to make that protein. So for the last part of these notes, we're just going to focus on transcription so we can see how that's working. So in order to make a copy of this DNA and rewrite it into mRNA, the DNA has to first open up, and then mRNA needs to be made. So it does follow Chargaff's rules, but remember, RNA does not have any thymine. It has uracil instead. And so when you are looking at that pink colored mRNA that just got created, where there would have been a T, it got replaced with a U instead. So you can see the DNA here starts out with TAC, and that got matched up with the mRNA that reads AUG. So it's still complementary, but instead of a T, there is a U. And then once the mRNA is made, it leaves the nucleus and it goes to find a ribosome, which is where the protein's getting made. While the mRNA is still in the nucleus, it does get kind of edited a little bit. There's things called introns that are in the mRNA that are going to get removed before it leaves. And then there are parts of it called exons, which are the parts that are good and needed to make the protein. And those are allowed to exit the nucleus and move forward with the protein synthesis process. So when we're actually looking at the transcription process and how we are rewriting the DNA into mRNA, it's very important that you remember that DNA will always have T's in it because it has thymine. But when you are going to rewrite this DNA into mRNA, wherever there would have been a T, there is now going to be a U. So for this example here, we were transcribing the second strand of the DNA, and it started out as ATG. And so that got matched up with the complementary bases UAC. And then when we do this, when we're writing the mRNA, that's transcription, which means this is happening in the nucleus because that's where the DNA is found. Okay, so on your own, let's see if you can do this one. So we are given the first strand of DNA, and if you could come up with the second strand, it would be complementary, following Chargaff's rules, which means it does have T's in it. And then you need to come up with the mRNA, so it's complementary. So if the DNA strand 2 is being rewritten and it says TAA, what will be on the mRNA strand? So the T is going to go with an A. The A's, if it was going to DNA, it would have been a T, but since this is RNA, they're going to be U's. Okay, so you should be noticing here that in the RNA, there are not any T's here. Okay, so I believe this is our last slide here. So once our mRNA is made in the nucleus, it leaves and finds a ribosome, and that's where it's going to be getting read to make a protein. So that's really everything. Protein synthesis involves two steps. We are starting off with transcription. We start with DNA in the nucleus. We rewrite it into mRNA. Then the mRNA leaves the nucleus, goes out to the cytoplasm where it finds the ribosome, and then that is where the mRNA can get read and it's used to make a protein. There are a few more details that happen in translation, but we will focus on those next time I see you. So that is it for this video. If you have questions, make sure you are asking.